Welcome to another explainer. Imagine for a second if, when you got sick or just started feeling the wear and tear of old age, you could pull a real-life Benjamin button. Like you could just completely reset your cellular clock and start your life entirely over again as an infant. Sounds totally crazy, right? Well, for a tiny little hydrozoan called Turritopsis Dohrnii, which you probably know as the Immortal Jellyfish, this isn't some science fiction concept. It's a biological reality. So today we're going to dive deep into the genetic investigation of how this organism literally escapes death. We'll be mapping out the precise genetic triggers that make this mind-blowing life cycle reversal possible. To really anchor us in the science right off the bat, we need to talk about the cellular alchemy making this happen. The core mechanism driving this quote-unquote immortality is a process called transdifferentiation, or cellular reprogramming. So what is that? Basically, it's a process where a fully mature, specialized cell, just a standard adult body cell, literally transforms itself into a completely different type of mature cell. Now, in almost all the model systems we have, trying to induce this kind of transdifferentiation in a lab is incredibly difficult, and observing it happen naturally, especially over a short time, exceedingly rare. But for Turritopsis dohrnii, this extreme reprogramming is just a spontaneous, everyday survival mechanism. Now, you might be wondering, why are top marine biologists and geneticists so absolutely obsessed with this tiny jellyfish? Well, it really comes down to the quote you see here. The ultimate goal of human regenerative research is to replace damaged cells when we face injuries or just natural aging. Think about it. If we can figure out the molecular drivers that allow this jellyfish's cells to just spontaneously abandon their current state and become a totally new cell lineage, we might finally uncover the foundational genetic mechanisms of cellular stability. And honestly, that kind of knowledge could one day translate into absolutely massive, game-changing breakthroughs for human regenerative medicine. Okay, let's get into the mechanics of this biological time travel. To understand the genetics, we first have to understand the life cycle. Normally, a jellyfish starts out as a bottom-dwelling colonial polyp, just attached to a surface. Eventually, it buds off and releases a free-swimming medusa. That's the classic jellyfish shape we all know. Usually, that medusa reproduces and then dies. But when the medusa of Turritopsis dohrnii gets stressed out, physically damaged, or even just senescent, which just means deteriorating with old age, it doesn't die. Instead, it sinks, settles onto a surface, and physically morphs into this intermediate ball of tissue called a cyst. And then, over a super tight span of just 24 to 72 hours, that cyst metamorphosizes back into a juvenile polyp. It literally hits the rewind button on its own development. And that brings us to the absolute focal point of this entire genetic investigation, the cyst stage. You can really think of this cyst as a biological black box. It's this highly unique, temporary, and historically pretty uncharacterized intermediate phase between the dying adult and the reborn infant. So when researchers sequenced the RNA of these different life stages, they were hunting for exactly what happens inside that black box, because it's right there, in that incredibly brief 24 to 72-hour window, that the actual cellular reprogramming occurs. So when the scientists ran a differential gene expression analysis across the whole life cycle, they grouped genes with similar behaviors into clusters. Take a look at this graph. Notice that dramatic, sharp peak right smack in the middle? That is cluster 5. This specific cluster represents exactly 224 genes that show almost zero expression in the adult or the polyp stages, but they absolutely spike in activity during the cyst stage before dropping back down again. This cluster of genes is, for all intents and purposes, the organism's biological rejuvenation engine. So what exactly are those 224 spiking genes doing while the jellyfish is a cyst? Well, by analyzing their functional categories, researchers found they're essentially acting as a cellular cleanup and stabilization crew. We see a massive uptick in genes related to aging and lifespan, like serine racemase-like genes. We also see incredibly high activity in DNA repair and damage response. Basically, proteins actively managing cellular destruction so things don't go haywire. There's also a strong presence of genes controlling transposable elements to manage DNA integration. And fascinatingly, several genes that we canonically recognize in humans for cancer and tumor suppression, like BRCA1, are heavily active. They're making sure this extreme rapid cell division and reprogramming doesn't spiral into cancer. Now, finding genes related to DNA repair and lifespan makes perfect sense for an animal going through extreme cellular rejuvenation, right? But as the researchers looked closer at that cluster 5, they realized something wild. The most thrilling and totally unexpected finding of this entire gene expression analysis actually wasn't what they could identify. Actually, scratch that. It was what they couldn't identify. This research uncovered a massive, completely unmapped genetic mystery sitting right at the core of this animal's immortality. The crucial point here boils down to this number, 44%. When researchers isolated the top 50 most significantly activated genes in that rebirth cyst phase, they found that a staggering 44% of them are completely unannotated. They're novel genes. They have absolutely zero known biological matches in our current genetic databases. To put that into perspective for you, in the jellyfish's overall transcriptome, only about 20% of genes are unannotated. But in the exact cluster driving this cellular reprogramming, nearly half of the most active genes are completely unknown to modern science. Just look at this. The single most significant, most highly expressed gene out of all 224 genes in that cis cluster has a really catchy name, TGI_DN103197_C0_G1. Rolls right off the tongue, right? Its expression value is basically 0 in the normal polyp, 0 in the medusa, and 0 in the reversed polyp. But it shoots up incredibly high, exclusively in that cis stage. So the single most active gene, the primary trigger for this animal's transdifferentiation, has no gene annotation, no known protein assignment, and literally no identified function. It's a complete mystery that is somehow actively orchestrating the reversal of aging. Let's shift gears for a second and look at the end product of all this. The researchers naturally wanted to know what happens after the jellyfish successfully reverts. So these set up this brilliant comparison, a tale of two polyps, if you will. On one side, you have a colonial polyp that was born totally naturally in the wild through regular asexual budding. On the other side, you have a reversed polyp, one that just survived a ton of stress by melting down into a cyst and doing that whole genetic time travel thing. Now, from the outside, looking at them, these two polyps are morphologically identical, but genetically, it's a completely different story. These heat maps do an amazing job of visualizing that hidden genetic reality. If you look at the comparison of the reversed polyp with the wild colonial polyp, that stark contrast in red and green shows us that these visually identical organisms have radically different internal genetic profiles. In fact, out of all the pairwise comparisons the researchers ran, the difference between the wild polyp and the reversed polyp showed the most distinct, robust patterns of differential gene expression of the whole study. Their origin completely dictates their biological wiring. When we contrast the two stages functionally, it becomes super clear why they're so different. The wild polyp is basically just relying on standard growth pathways, like G protein receptors, to expand its little colony. But the reverse polyp? It's operating like a biological first responder. It's heavily enriched with genes involved in embryonic development and chromatin remodeling. It's using tools like matrix metal proteinases to actively repair and rebuild tissues during this extreme regeneration. So the reversed polyp isn't just casually growing, it is actively repairing and reestablishing its entire self. Building on that, let's map out the larger picture of the organism's transcriptome. Look at this Venn diagram of the shared and unique transcripts. See that red circle representing the reversed polyp? Even though it shares over 16,000 transcripts with the wild colonial polyp, the reversed polyp retains a massive 9,729 unique transcripts that you won't find in any of the other life stages. It's just staggering. That huge number of unique transcripts really drives home just how distinct this regenerative pathway of reverse development is compared to normal, forward biological growth. Which ultimately leaves us with one massive looming question. What are we going to discover when we finally unlock those unknown genes? Turritopsis dohrnii is giving us a living, breathing, easily observable system of cellular reprogramming happening right in front of us. As researchers keep sequencing its genome and slowly start to annotate that 44% of mysterious, unidentified genes powering the cis stage, we are standing on the edge of an entirely new frontier. Could mapping these novel genes actually teach us how to control cellular stability? Could it completely revolutionize human regenerative medicine and change how we treat aging forever? The blueprint for immortality might literally be written in the DNA of this tiny jellyfish right now. We just have to learn how to read it. Thank you so much for joining me for this explainer, and as always, keep staying curious.